Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I'd like to invite you to mark your calendars for the very first Dr. GPCR Virtual Cafe held on March 26th, starting at 11 a.m. EST. Our first live virtual cafe guest is Dr. Brian Airy from Bristol Myers Squibb. Space is limited, so don't forget to get your tickets today. Visit drgpcr.com/virtual-cafe for more information. To present your original work or your company's technology, or to sponsor a Dr. GPCR Virtual Cafe edition, please fill out the form on the cafe's main webpage. The goal with the Virtual Cafe talks is to bring you GPCR-focused live presentations every month. You'll be able to join us live and interact with the speaker and attendees. We hope you'll join us. To stay on top of all your GPCR news, subscribe today to our monthly Dr. GPCR newsletter at drgpcr.com/newsletter. Last but not least, find us on YouTube. You can subscribe to our YouTube channel to watch the latest Dr. GPCR podcast episodes and much more. Hello, everyone. This is Yamina from Dr. GPCR. I'm delighted today to have Dr. Elva Zhao with me this morning or this evening, depending on where you're listening. Elva is a research fellow at the Monash Institute of Pharmaceutical Sciences, Drug Discovery Biology in Parkville, Australia. And I'm very excited to chat with you today. Hi, Elva.
Hi, Amina. Thank you for the introduction. Hi, everyone. Good morning, good afternoon, good night.
Exactly. Yeah, I didn't realize that, you being in Australia and me being in Boston, there was not only 12, but 13 hours of difference between us.
Yeah, but thanks to the magic of internet, I guess.
Exactly. You can't really tell. I could only tell by the fact that it's dark outside where you are.
Yeah. And then my dog is sleeping.
Absolutely. Can you a little bit introduce yourself to us and talk to us about the work that you're doing?
Yeah, sure. So I was born in Shanghai, in China, and I did all my undergrad study and everything, all the basic education in China. And after I graduated, I moved to Canada. So I did my PhD in London, Ontario, so Western University, or it was called University of Western Ontario. So under the supervision of Professor Peter CDX. So the PhD work that I was doing is actually not on GPCR at all. It's not on the receptor. So it's more on the regulation of G protein signaling. So how the G protein cycle is regulated by different modulators, that's intracellular modulators, such as RGS proteins and GPSM or AGS protein, local motif containing protein, all the small molecules that can regulate the kinetics of G protein cycle. And then after I graduated, I moved to Melbourne. So working in MIPS, so Monash Institute of Pharmaceutical Sciences and Drug Discovery Biology. And I was working with Professor Nigel Barnett for a couple of years. And so the work in Nigel's lab was focusing on a family of receptor called protease activating receptor. And then in 2016, I joined the group that co-lead by Denise Wooten and Patrick Sexton. So it's mostly focusing on Class B GPCR and the bias signaling of Class B GPCR. And one of the receptors that I spent most of the time with is GLP1 receptor.
Wow. And what made you decide to join Peter CDX Lab in Canada? Because it's still a big move from China.
Oh yeah, for sure. So I guess the idea of going abroad or overseas for postgraduate, it was sort of implanted in me since I was little, because both in my background, so it's more like both my grandparents, my grandma and granddad are both working in science, and they were in China. So my grandma was a pharmacologist, and funny enough, working on diabetes, which is the similar area that I'm working on right now. I don't know whether there's some deep down connection or not. And my grandpa is a toxologist. So I was always in their lab, just like, play and stuff like that. So my university undergrad is actually a small university in northeast China, and is a pharmaceutical university. So the major is sort of within the pharmacology or biochemistry, that field. And then going overseas, I think it's really my family is encouraging me to not only to pursue my study, but also to experience more of a different lifestyle as well,
Yeah.
and to be exposed to different diversity and diverse science. And then at that time, I didn't know I'm going to join whose lab or whatever. So it's more, so to go into Peter's lab is actually more of a, I don't say coincidence, because as undergrad in China, we don't really have any deep down understanding of any particular scientific field. So it's more of which uni choose me and who gave me the opportunity to do an interview. And
Got it.
it's not really direct by the science, but I think after the chat with Peter, we feel like, okay, he's a super nice guy, and I feel like I can have a good time in his lab. So that's how it brought me in this whole GPCR and G protein world.
It's so interesting that your grandparents worked in science, and you spent time in the lab. I can see you racking tips or doing things like that.
Baby me, just there following them around.
in the lab. And so you could say that it's almost kind of a family tradition to be in science, and you got inspired by your grandparents.
Exactly.
Which is phenomenal. So tell me more about your first contact with GPCRs, or with G proteins, or with the signaling field.
So I think the first thing joined Peter's group is, so the first thing, I wasn't introduced to any of the receptors. So because Peter's lab back then was focusing on RGS proteins, and how RGS protein is regulating the G protein cycle. So I think my first contact point is actually how the G protein cycle works and what are the rate limiting steps and all that kind of thing. So my primary point is to understand how GDP dissociation, GTP binding, and GTP hydrolysis is regulated. So it's really like basic and fundamental question on the G protein cycle, and then we did study or use receptor as a tool to study. Like we have this membrane assay, so we can measure GTP hydrolysis, we can measure GTP gamma S binding. So we want to look at it in the presence and absence of different modulators, different RGS protein, different go local protein, GPSM protein, to look at the outcome. So actually the first receptor I'm in contact with is muscarinic receptor. But I have absolutely no idea what it means and what it does and like where it's expressed, because that's not my focus of the study. It's more on how the G protein is cycling around.
Exactly. But muscarinic receptors, I think it's a great model, and ligands are cheap. It's great to work with. It's a great model receptor.
Exactly.
I think a lot of people are not necessarily looking at a specific GPCR, but more at what happens once the receptor gets activated, like the muscarinic receptor, beta adrenergic receptors as well.
Yeah, those are like the prototypical receptors.
Exactly. When you know that it's a DNA in some tube, and you just add it to your cells, and it should work all the time.
Yeah, exactly.
That's great. So can you tell us a little bit more about the transition from China to Canada? I remember we had discussed this in an email while we were setting up this interview, that you going from a full Chinese 24 hours a day to an English environment. How was that experience?
It was quite an experience. So at the beginning, I thought, because a lot of people are going overseas to study, and I have to say, you have to be the student to actually know what the feeling is to be an international student and to be an international PhD student in a foreign environment, and to try to speak a language or study in a language that you never confidently spoken before. So my personal experience when I was in China, so we did study English, like from, primary school and all the way through university, we did study English. But in terms of study English and speaking and listening and interacting, hang out in English is completely different things. And it's more of a challenge when you actually experience it. So this is one of the stories that I actually talk about during one of the diversity and inclusion week that Monash, our MIPS hold a couple of weeks ago. Is the first thing I moved to Canada, there was a molecular biology course that every new master student or PhD student were signed up to. So it's basic. It's very, now you look back, it's very easy. It's like to teach you how to do molecular cloning. You have the restriction enzyme, you cut it out, and then you put it in a vector, and then you run a gel, DNA gel. And then there's a little quiz toward the end of it. And I was doing the quiz when I was answering the question. And that was like the first week or the second week. I just landed in Canada. And I couldn't understand a lot of the question.
Wow.
Just I cannot. So, and now it's like one thing stick to me, and it's really like wowed me, is that there's one sentence about, okay, how are you going to detect your DNA? And then, like you run a gel or you have a gel doc, whatever. I don't know the meaning of the word detect.
Wow.
So that was one of the things that like you need to experience, and you need to use the language. You need to use the language frequently enough so that you can register. So I know the meaning in Chinese back then, for sure. But the two language didn't really click.
Well, it's two languages that are very different. So I can relate to it
a little bit.
Yeah, I can relate to it because just to give you a little bit of context, I do speak 5 languages. But English was the last language I ever spoken, and ironically, my mom is a French and English teacher. But not that she never wanted to, but I never wanted to, learn English from her. And it's not the same thing as having to use it on a regular basis.
Yeah, exactly.
So I was in Canada as well, in Montreal, so in Quebec, in the French region of Canada, and I would watch TV in English, but I never had to speak English until I went to a Gordon Research Conference, and I was sharing my hotel room for the week with an Italian student who was also doing her PhD in Canada, but she didn't speak French.
Right.
So and for some reason my brain just clicked, and that's when I started just speaking English. But up to today, there are words that I know in English what they mean, but somewhat they don't make sense to me. I know what it means, but it just doesn't make sense.
Yeah, exactly. So I think that's actually a similar situation. So for that course, I actually at the end I have to audit instead of, because there are so many things I have to catch up on to bridge the gap in terms of language.
Yeah.
And then I think in addition to the English barrier or the language barrier is, a lot of cultural differences. Because like for us or for me, I might be quite intimidated or shy to begin with. And I think it's actually might be common for a lot of international students, not only Chinese students, or a lot of international students whose English is not the first language, or whatever the language is not the first language that you study your PhD, is that at the beginning when you're talking to your PI or talking to your supervisor, and when they are explaining something to you or they're asking you tend to just nod and say yes and not sure what you're saying yes to, and then as a supervisor, they will think that you understand because of your response. But in reality, you don't really understand what it is. And then once the moment passes, it's too late to ask.
Yes. And then you can't go back and say, actually, well, you don't feel comfortable going back and saying, actually, I didn't understand what you were saying.
Yeah. And then at the end, I think there will be frustration on both ends, because if you ask, the supervisor normally will be really nice to try to explain in a different way and everything. But for us, we're just too either scared or shy or don't want to show that I don't understand.
Yep.
We just go with it. And then there's this, so that took me a while to sort of adjust to the fact that if I don't understand, I need to say it, because that's going to cause trouble on both ends.
Yeah. Do you remember the moment when this thing clicked, when you were comfortable enough in English to say, hey, Peter, I don't understand. Can you explain it to me differently?
Yeah, because I think that's something that I really have to say thank you to Peter and to my mentor, because I think Peter is one of those guys that's extremely patient. And I think he's quite sensitive, or he's quite understanding that as an international student, you might have this kind of problem. So at the beginning, maybe where I'm trying to say yes too much without knowing, and then he's just assumed that I understand. But there's nothing, like there's nowhere to hide, because if he's asking you to change something in your essay or in your report, you didn't do it, and he's gonna be like I told you, or you said yes, and then after this discussion, he's like, Elva, so it's just, if you're not sure what I mean by this, I'm happy to explain, and I'm happy to say it again, and I'm happy to just explain to you until you understand. So I'm like, okay, it might be okay just to say that I don't understand.
Yeah, it's funny because I think the issue, or the situation that you're describing is not, it's at multiple levels. It's the fact that you're an international student, the fact also that you do not have the experience that your PI has, or your mentor has, and you definitely don't want to look not knowledgeable or incompetent to say that. So I think I've done that too, saying yes, although I perfectly understood what I was told, and thought to myself, it's okay, I'll figure it out later. I'll take notes and I'll try and figure it out later. So I can only imagine, adding on top of that insecurity as a new student, as a PhD student, the language barrier.
I think another thing is to add on to this, to don't want to sound not knowledgeable or not trying, or is the confidence.
Yes.
You don't have the confidence to say that I don't know.
Yes. Yeah. And it's normal. And that's something I learned quite late, but it's normal. No one is born with confidence. You build confidence over time.
Exactly. And it takes time. And I think everyone, like, the confidence issue may or may not be related to the fact that you're international or not, because with time, when I'm like interacting with different students now, and then different colleagues and people from different backgrounds, I feel like everyone goes through similar paths in terms of confidence building and all that,
Yes. And if I can also add, I had this discussion with one of my colleagues. I wonder if the confidence, you gain the confidence with time, you get exposed to new things. And I have to salute and applaud all the international students who have that, that initial confidence to actually get out of that cocoon and their known environment into a new environment. But I wonder if it's also a gender thing. I think, I don't know. I feel like guys will also have the same issues, but maybe they won't show it as much as I would have done as a female.
I think it's like what you said, it's like multiple layer.
Yeah.
So there's the cultural, there is the nationality, there's the language, there's the gender, and then you have the combination, and there's the matrix where you just lack that confidence, and where the source of your lack of confidence is coming from.
Exactly. The thought that came into my mind while you were mentioning this is when you measure function of GPCRs, and at the end you have all these pharmacological data, and you come up with that spider web representation for the bias. I think that's
the web of bias.
Yes, I think the web of bias changes from person to person when it comes to these things, but at the end of the day, it's just a combination of things. So you graduated from Peter's lab with your PhD. What led you to the other end of the world, in Australia?
For that, because Australia was not on my radar or on my back. So when I was applying for postdoc position, I applied to a couple labs in Canada, couple labs in the US. And so got a couple interviews and things like that. And at that time, I think that was 2012. Yes. At that time, there was the first or the second of the MPGPCR meeting was held in Melbourne. So that's every other year. And it continues. And so Peter was invited to that very meeting. And at that time, MIPS or DEB was sort of just moved to a new location, so to the current Parkville campus. And so the facility is quite new. And when Peter travels back from Melbourne, so what he told me is that, have you considered Australia? I'm like, I don't really know, because never. And he's like, well, if you want to work on some GPCR, there is a place that's just focused or centered in GPCR related work. And then, you know, there's Patrick, there's Arthur there, there's Roger. And he's like, the facility is great. And what's most impressed him is that the science here. And all the people here are really, really passionate about what they were doing. So I'm like, okay, I will check it out. So, and also on top of that, my auntie is actually living in Melbourne. So that's sort of like also part of family here. And another thing that's actually tricked me to move to Melbourne is that on the globe, you feel like Melbourne is closer to China, but not really, because you have to go through a different direction. So it's actually similar length compared to Toronto to China.
Wow.
But all the combination actually just then, and here I am.
That's fantastic. Before we dive into your postdoc experience, can you tell us a little bit more about what motivated you to do a postdoc? And a little bit more detail. So you mentioned that you sent out emails to scientists in Canada and the US, and then Peter had suggested to check out Australia and the team in Melbourne. But for young scientists who are listening to the podcast, what was your thought process
in terms of
making a decision to go for a postdoc?
Okay, so I think for me, the center of my logic or my thinking process is always, what do I want to do? So I really enjoyed so I started my postgraduate as a master's student. So, and then 1.5 years into the program, I transferred to PhD. So I think during the 5 years or 5.5 years doing the PhD, it really like opened up my curiosity for science and for G protein and understanding all these things. So toward the end of the PhD, I was like, okay, so I do have the option, you know, I either go to industry to do a postdoc in academia, to move back to China, and, you know, all those kind of things. So at that time, I just feel like I'm not done yet with answering my own question. And also I always have chat with Peter about different options and what, so Peter is always like, so don't worry about, you know, whether it's industry or academic, and is there a question that you want to answer, or is there something that you want to work on, and then we'll figure out whether there's an environment that can help you to do what you want to do. So at that time, I wanted to know more. So I'm not finished with the G protein. So I want to know more of how it's regulated, but I want to know more of upstream. So I want to know how the G protein got activated. So receptor is a natural choice. So I want to look at how receptor activating. And then at that time, bias agonism or bias is relatively new or relatively novel concept. And I think all this thing just lead me to say, okay, so here's something, it's a bias talking about GPCRs, and it seems interesting, and I don't understand. I do want to understand more about this, and I'm fresh out of PhD. I don't hate science yet, So why not do a little bit more? So that's my thinking process.
That's amazing. And I loved you made a couple of great points that I want to highlight. So one, you considered the different options that there were out there, industry postdoc or academic postdoc. And at the end of the day, what you focused on is really where your interests lied. And your interests lied in going further and understanding more and trying to solve a problem that you were passionate about.
Exactly.
And
I think like, so I was the one that chose my interest, or like sort of knowing a little bit of the general direction where I want to head it into. And I have friends who also did PhD in either the same university or different university, and during their PhD, they know, okay, so maybe a academic life or day to day in the lab dealing with, well, a tube of water with another tube of water is not their thing. So they started to look into, science communication and consultant, or, other area of clinical trial. And that's all fine. It's just different choice. It's just different options.
yes, I think, and I think it's a great point that you make again. You can do pretty much anything you like with a PhD. You do not have to continue working in a lab. You do not have to become a postdoc. You can do pretty much anything you like. I think the most important thing is to think about where you want to be, what you want to do. Talk to people who have done different things with their PhDs, and then make a decision out of there. And if by any chance you make the wrong decision, it's okay.
Exactly. I think another thing is, I think it's quite important, and in this I become realize of this is that you need to make your decision. It's not what other people think you should be doing, or okay, so I have this PhD. If I do this, but not this, is that a waste of a degree or something? It's your life. It's not about you make what a degree makes you. So, and then you're going to regret it if you say, okay, so I think I should do this because I spent 5 years on this, although I hate it, but I have to continue or something like this,
Exactly. And I didn't think about what I wanted to do when I finished my PhD. All I knew is that I loved working in the lab. I loved GPCRs, and it just seemed like a natural thing to go and do a postdoc. And it was the best 5 years ever.
Yeah, exactly.
Especially living in New York City. So I think that also was an added to it. But yeah, if I could go back in time and give myself advice, I don't think I'd be doing something different or differently, but I wish I had somebody to tell me, hey, just think about it. Where do you want to be in 10 years? And go explore.
Yeah, and I think the good thing about, having a great mentor that will support you is that, like Peter, I think he has an idea of what he thinks, but he never pressured that idea onto me. So it's like, okay, so here are all the options. What do you want to do? And I can sense that he has that hint, but he never put that on. And like, I feel like you should do this or you should do that type thing,
Yeah, I think that's valuable. Just he was guiding you in your thought process and letting you.
Still guiding me now. Like I still have interaction with Peter and here and there. I'm more like a collaborator and all that kind of thing,
That's phenomenal. That's phenomenal. So you moved to Australia, and then you decide to work on GPCRs, PAR receptors. Tell us a little bit more about your experience as a postdoc in Australia, again, in a totally new environment.
So I think moving from Canada to Australia is actually, like, Canada, the society or the feeling wise, Canada is quite similar to Australia. And the differences that personally I experience is the work environment, is because I think the way we operate in DDB or in MIPS is more of a collaborative way. And then we all in a big center or central lab that a lot of people are sharing the space. So you can just interact with scientists with all sorts of different expertise. And then because for DDB, it's actually quite GPCR oriented, or at least at that time when I first joined, So, and also we have a big, structure side, and to look at structural biology of GPCR, and there's a huge focus on pharmacology of GPCR, and there are people working on, class A GPCR, class B GPCR, class C GPCR, and there are people working on in vitro pharmacology, biochemistry, ex vivo, and in vivo. So that's more of a eye-opening experience for me. So it's more like when I first joined DDB, it's like, okay, let's collaborate, and then whoever you want to talk to, you talk to them. And then I have a couple moments where I'm like, I don't know how to analyze this data and how to calculate the web or how to do whatever thing, and I don't know who to talk to. So I just bring my laptop, go to level 3 or the lab where a lot of the lab heads have their office. So I'm just like cruising around and to see who I can bump into and start talking to them. So that's the experience that you interact with them, and it's very collaborative, because in Canada, at least at Western or in our department, it's more like, each PI has their own lab, and then it's more focused or centered on their science. And in a department, like for me, I was in Department of Physiology and Pharmacology, and it's a lot of diverse research going on. There might be people working on sepsis, working on, hypertrophy, and then there's pharmacology lab. But for here, I think it's both bad and good. It's so focused on GPCR because it's really receptor pharmacology, but we don't really have the diverse on, a lot of ion channel or all the tyrosine kinases and all that kind of thing is lacking. But if you want to study GPCR, like you just go there and then you talk to people.
It's like GPCR for GPCR heaven for GPCR fanatics.
Exactly.
That is awesome. And how did you, so what you're working on PAR receptors, did you like it? What did you learn out of it? And how difficult was the transition from working on the GPCR activation cycle, G protein activation cycle, and moving one step further up towards the plasma membrane?
Yeah, well, so I think at the beginning, it's like, so the PAR receptor is working with Nigel. So I worked with Nigel for about 4 years. Mm-hmm. Yeah, a little bit over 4 years. And so the PAR work I did is also focused on bias signaling of PAR and PAR2 receptor. And then including the work I'm doing now with Patrick and Denise is on bias signaling of GLP1. So to moving one step up is you know a little bit during your PhD, but you don't know the full story of how. Now I'm more focusing on, especially the GLP1 work, I'm more focusing on how the ligand is engaging with the receptor and how the receptor is engaging with the G protein, that type. And then, so the work that I did with Nigel and also a couple of years with Denise and Patrick is on, okay, so how ligand binding triggers this different conformation of the receptor and triggers more on the downstream signaling. So I kind of bypassed the G protein. So I bypassed my PhD. So I studied the ligand binding to the receptor and how it's activating, cyclic AMP and calcium and ERK and all that kind of thing. So what I started to do recently, not recently, like from maybe last year or something, is try to bring my PhD back into the picture. So more to study the transducer coupling. So it's how the receptor is interacting with the transducer or the G protein. And what I want to study in the future is how this whole thing is regulated. I will try to bring in, RGS stuff and GPSM stuff, and then put that in a more physiological context.
Which is absolutely the way to go if you want to, translate the basic research into something useful and hopefully get to discover drugs or to discover something that might help patients in the long run.
Yeah, exactly.
So if you had to choose between PAR2 and GLP1 as your favorite GPCR, which one I have to ask this.
Okay. So yeah, I saw that on the thing that you sent me. It's like, what's your favorite GPCR? It could be a family of receptors. I think well, GPCRs are GPCRs. They're all my babies, and they're all good. But I invested a lot of effort or lately on Class B GPCR. It might be because I work with Denise and Patrick and using GLP1 receptor as the receptor that I study with, I started to introduce transducer coupling to it and try to understanding how this works. So I feel like for me, I know GLP1 a little bit more than other receptors. But if you ask me what's my favorite, let's say, signaling component, it's still the G protein side. So I know GLP1. I really, really like GLP1. And it's a lot of story. I feel like it's this thing that has a lot of story can build around it. And I started to understand it, but it's still mystery. And the mystery part is actually the transducer part. It's the G protein and the regulation part.
That's great. Yeah, it's funny. I had the podcast episode with Dr. Silvio Goodkin. I always ask this question, and then Silvio had mentioned that actually his favorite GPCR related signaling molecules were G proteins, but he made the analogy that he has two daughters and it's very difficult to choose between both daughters which one he loves more. So it's kind of similar with his G proteins.
Yeah.
At this point. So I love the fact that the PhD work that you've done comes back into focus, and you're kind of marrying it with working on GPCRs and on GLP1. So you mentioned that your grandma was working on diabetes, and you alluded to GLP1, and going for a circle family wise and working on diabetes. What can you tell us about GLP1?
Okay, so basically GLP1 receptor is a glucagon-like peptide receptor. So that's a receptor is very important in regulating glucose homeostasis. So it's a very attractive therapeutic target for type 2 diabetes and obesity. And lately there's more and more evidence suggesting the central role of GLP1 might also be contributing to its potential target for neurodegenerative diseases such as like Parkinson's disease or Alzheimer's diseases. So it's actually a quite hot target on a lot of different things. However, the endogenous ligands for GLP1 receptor, which is mostly GLP1 and also oxymodulin, are quite poor therapeutic candidate because it's short half-life, so it's quite short. So it can be digested by DPP4 quite fast. So one of the focus that was from the pharmaceutical side or for the science is to extend the half-life of a peptide mimetics to target GLP1 receptor. And then lately there is also more suggestion or strategy on developing novel peptide on targeting more than one incretin receptor, such as if you target both GLP1 receptor and GIP receptor, another very important incretin receptor, even together with glucagon receptor, to try to bring up or take advantage of the therapeutic beneficial and to avoid unwanted side effect profile.
Wow. So you mentioned the so the problem with GLP-1 peptide in the context of diabetes is the short life. I did some digging previously, and there are multiple drugs on the market that target GLP-1 in the context of diabetes. Actually, there's quite a few.
There's 6.
Yeah. So which is better than for other GPCRs. So what is there that what do we still need to study? What do we still need to understand, since there are 6 drugs on the market?
I mean, like, by no means I'm the expert on this because there are big names there, but my understanding is that out of this currently approved GLP1 receptor agonist, the clinical benefit was there, but the side effect profile is one of the major things that hold off So, for example, one major thing for GLP-1 receptor agonist is the gastrointestinal side effect. So you might have vomiting, you might have, nausea and all those things. And that brings it back to, how important it is to actually understanding how this receptor works and to try to come up with a profile that can link the clinical benefit effect versus the unwanted side effect and try to develop this therapeutics that's more focused on the beneficial effect compared to the other one. And also because it's a peptide, the oral availability is a problem. So right now, in terms of the peptide ligand, there's only one that is orally available, which is semaglutide. So that's just been approved either this year or last year. All the others, you have to seek other administration route.
Okay. So they're injected most likely.
Yeah.
Wow. That's great. So and how does your day look like? I'm assuming, yes, your day, your regular day right now working on GLP-1 with Denise and Patrick's team.
Well, you mean like my day this year or my day regular in last year? Because it's quite different.
both. So I'm assuming, it's not the same type of day that you would have as a PhD or a master's student, and now as a research fellow. How was your day before COVID, and how is your day during COVID times?
So my day before COVID, is actually so I started working with Denise and Patrick the end of 2016, the beginning of 2017, and so I was lucky enough to get a main focus of my own project and also co-supervise a couple students. So in our group, the way we run is that we have regular meetings with students. So every fortnight, so every 2 weeks, so we have meeting. So and both Denise and Patrick will always be there for the student progress meeting. So if you're talking about typical day, so I might have, one maybe average one student progress meeting and talk about how student project works. And I will have, experiment day. And I was extremely lucky to have a very, very nice, very good research assistant to help me to plan out my experiment. And lately, I've been more focusing on developing new or set up new system or new assays, and then supervising students to do more assays, different bread assays, kinetics, and all that different kind of thing. And then they will be having, so this is before COVID. So I normally will have, toward the end of the day, I will try to find some time to run. So because I exercise and try to do 2 or 3 times of running every week. And when I got home, it depends on the day. So normally after dinner, I will either try not to bring work home, but that may or may not always come true. So it might have a period of time where, whether it's a funding application time or it's fellowship application time. So during those times, the day might be merged a little bit where you have to focus more on the writing and here and there. But I try to have some time at night toward the end of the day to just relax. Yeah.
I love that you brought this up. I didn't even think about asking that because I think so work-life balance is very important. Mm-hmm. And when you're a PhD, you're most likely young and energetic, and you just want to spend as much time possible as much as possible time in the lab. But having that balance, how do you think that balance, that the fact that you're running and the fact that you try not to take work home benefits your work as a scientist?
It's extremely important. So I think the day I described to you is probably the day last year. So if you ask me about 2012, it might be a completely different day. So in 2012, I might be going to the lab at 8 in the morning, start the experiment, and all the way skip lunch, skip everything till like 6 or something, and go back home and quickly eat something and just feel like I need to read more papers, analyze the data, and then think about stuff until late. So but you have a time where you just burn out, and it's really like when you are in the state of burning out, it's really hard, or it's harder to bring yourself back up. So and then lately, after this different cycles of, you try really hard and you're exhausted, and then I sort of like come up with, I know my own tempo or my own routine of, okay, so I need to relax, or I need to have some work life. I need to have a life, basically. I need to have something that is not work related, and that really help you balance out your energy, and that will actually benefit you to work more efficiently during the time that you're actually working. So I didn't run at all when I was in Canada. Like, well, it's cold.
I was going to say that. So it's not because of the winter; it's because you were working in the lab. You can still run in Canada.
Yeah.
Most of the year.
Exactly. So, and also during PhD, I was like always just, and I feel like I need to do the work. I need to do all these things. I was like constantly want to ask Peter questions, and there's sometimes Peter will work late. And there's one time I remember, sorry, we were discussing data and things, and I was asking him all these questions, and Peter is like, "Elva, it's 10:30 p.m. Can we talk about this tomorrow?" So I think he's like half asleep or something like that. And then here, I learned to just adjust my time and then put the energy in the work during work time. And then it is okay to work overtime, but that shouldn't be the normal case. And I developed this habit of running when I moved to Australia as well. Is because some of the colleagues, and they were signing up this like running event.
Wow. Yeah, I think it's important to hear what you're saying for trainees, because yes, and it's good to be doing the work and it's fulfilling. And I think as scientists, we all need that dopamine rush whenever something is working in the lab and you finally get an experiment up and running. But then again, it's a marathon, it's not a sprint.
Yeah, exactly. I think we all have this feeling that, okay, so right now the experiment is working. There comes in waves. So this period of time is always working, and you never know when that wave is gonna go, and then like nothing will work. But I think maybe it's because I've in this for a long time. I know it comes and go, and sometimes you get better control over it because you kind of know what you're doing, but you don't always have control. So what you always have control is how you distribute your time, and to have some downtime, to have some relaxing time. So for me, for the weekend, well, I'm still able to go out because in Melbourne it's kind of in lockdown right now. But I always have like a day that is not work related. So I go out to run in the trail, run in the mountain, run somewhere, and just hang out with friends. And with non-scientific, like with friends that's not in the same community, and friends from all over the place, and just try to, shake it off a little bit, and then come back to the lab again the new week.
Yeah, I think it's really important to have that, that downtime from the lab, because, and I said this multiple times, I feel like I had the best ideas while not working in the lab, while doing something else, and that's when your brain has the time to process.
Exactly. So that's something like I was chatting to one of my best friends when I moved to Melbourne, Tinny. So she did her PhD in Johns Hopkins, I think, and then she moved to Melbourne to join NIDO's group as well. So we were chatting, and I was during a very stressful time, and then I was like, I feel like I need to do more. I feel like I'm not working hard enough. I'm not productive enough. All this kind of thing. And I'm like, what are you doing? And he's like, oh, I'm just shopping. And then she's like, it's not like your mind switch off, because as a scientist, your mind is always on, so, and he's like, just come and just do some shopping. We might have some ideas during the chat. then you never know when the things just click and you're like, okay, I might have to do this, and it might be you're just like picking a pair of shoes or something. So you don't have to always physically in front of your bench or physically in front of your computer to do that.
I think so. And then walking the dog is also a nice way to disconnect.
Yeah, for sure. Once I have Tillie, that's just, so Tillie is, so we adopted Tillie 4 years ago. And then because of Tillie, so every day I have to walk her.
Yes.
So no matter how like lazy you are and you don't want to go out, you have to, because she's there just barking along saying that it's my car walk.
Exactly. And it's her time.
Yeah, exactly.
It's her walk time. As I mentioned before we started recording, I also have a dog, and his name is Apollo, and he is really protective of his time. He doesn't mind waiting a little bit to take him out, but whenever we go on a walk, it is his time and it is our time together, and that's such a great way to disconnect, because you're just focusing on the walk and on the dog.
And then I think it's funny for dog or for Tillie is that for some reason I feel like she knows it's time to walk. She knows the time of the day. So during COVID, so we suddenly have a lot of time at home. So before she was always in the backyard, and now we always have it's like Tillie's in heaven. So every day when I'm like sitting in front of my computer, either doing meeting or writing paper and all that kind of thing, when the clock hit around 4, you can see Tillie is like sleeping right now. But if it's around 4, you can see her just shake it off and start walking in front of me, and then just stretching here and stretching there, and then start wagging his tail and start barking if I try to do some more work. And I'm like, okay. So that's a physical break.
It is.
As well. It's really nice.
It is. I might say that, people who will be listening to the podcast episode. So right now we're in October. It'll air in January. And I was talking to you, and through all the conversation, Tillie has been sleeping like a baby right behind you.
Yeah, actually, like because of this COVID and the Zoom meeting, there's one time we have like multiple meetings along the day, and toward the end, my colleague, oh, she's alive. Like, yeah,
she is alive. I saw her stretch a couple of times. So we talked about your typical day before COVID, and a little bit about how your life is right now with COVID. So you spend a lot of time at home writing papers, having Tillie guard you, making sure that you're doing your work during the day while she's sleeping,
or having the break when I have to have the break.
Yes. You have a live schedule monitor who offers a lot of love, I'm sure. Any other changes that came with COVID in your life? How are you coping with working from home?
Yeah, well, surprisingly, like I wasn't expecting this type of coping at all when I first starting to think, okay, I might actually spend a lot of time in lockdown or in isolation. So very well. So I think over the years I starting to get into a habit of adaptation. So adapt to, maybe it's because I change environment a lot relatively. So I adapt to the environment and I set up new routine. So for me right now during COVID, so because in Melbourne we started sort of first wave and second wave, so the lockdown has been going on and off since April, and now it's like mid-October, so it's like literally 6 months. So but during this 6 months, I started to, my typical day during COVID is I wake up and shower and everything, but I will do 1 hour or 1.5 hours of reading my novel or books before I start working, and that has been really, really relaxing. And I read so many books that I feel like I left out all this previous years. And then and then during COVID, I finally got a chance to actually organize a lot of the data that has been accumulated over the years to put them in a package that might potentially become a manuscript soon. So those are actually the things that I couldn't do before COVID because I feel like I need to go to the lab. So I think that's something that in a stage where I'm sort of a little bit starting to thinking about packaging my own paper and writing paper and managing this project is I need a big block of time to think about the project rather than focusing on generating the data. So COVID actually gave me a chance to think a lot about my project, about my data, and about the project that I'm currently doing, as well as the project that I want to be doing in the future. So COVID also gave me a chance to start set up new collaborations with different people and go to different conferences and from different time zones. So and I think routine, like changing the routine to adapt the current environment is very, very important. And I started regularly exercising, like every day, go for a run and go for this. And I start, because I'm doing so much work at home and save on the commute time to go to work and to go home from work, that actually saves about like 1.5 hour, 2 hours every day. So I can actually afford to, do some cooking and learn different skills and try to play a little bit of piano and watch movie and all that kind of thing. So actually, it has been great.
Yeah, I think that's important. And although it's a pandemic, it's definitely no fun for anyone. But I think as humans, one of our fortes, although it comes with pain, is adapting to new realities. And I could only echo the fact that I think I've been to more virtual conferences and learned more this year than I've ever done, on a yearly basis, because I usually used to go to one conference a year or maybe two, depending on where it was.
One of the things I did over the COVID, and I am quite pleased or quite proud of, is I was chatting to Peter and we did a lab reunion over Zoom. So it's the lab, so all the lab mates when I was in Peter's lab. And so people are now in Japan or in the US, in Canada, and in Germany, and all different places. And a lot of them I haven't seen since 2008 or maybe 2012. And everyone joined at the same time, which is phenomenal. And that's actually a great 3 hours. And because of the internet, because of this thing, so we'll be able to communicate with each other.
I think it pushed a lot of us to slow down.
Yeah,
just
and to
down.
Think.
And
there's
yes,
more to think, to read about stuff.
Yes, and then focus on what's really important on the long run. So we've talked a lot about some of the advice you have or for trainees. Is there anything else, as any take-home messages you'd like to share with trainees to get them to get them through a PhD or through a postdoc and still help them get that balance that you need to be a great functional scientist and not burn out?
Yeah, I think like what you said, doing science is a marathon. It's not a sprint. And also there's something that I think is very good advice, is that to be a scientist, you need to stay curious and to be always imaginative about things. And so that's something I always keep in mind. Just be curious about everything and stay passionate about the things that you're doing. And really in the core, knowing what is the scientific question that you want to answer, and what is the thing that, and then focus on that question. Don't try to be distracted by, I need a big paper and I need this, I need that. I mean, the reality is you probably need something to build up and for you to secure some of the funding, but always just keep the core to what is the question that you want to answer, and then And then also another thing I think is very important to know that it's okay to feel down sometimes, and it will come back up. And over these years, one thing I developed, or one skill I developed, is on the good day, quote unquote, relatively okay day, I always remind myself of the not so good days, thinking that, okay, so I've been there, but I'm here now, so I can do this thing. And so this I think this really helps you to keep your mood in a balanced mode. So you don't have this dramatic roller coaster. And then over this long years of doing science and in this thing, it comes and go, and then it's up and down, and it's okay. And then talk to people. No matter what you feel good, you feel bad, you feel sad, you feel happy, always reach out, because you'll be surprised how much help you can get if you just ask.
Yes, and then if you just externalize whatever is making you sad or happy, I think you can get into this loop of problems or overthinking in your brain, and just saying it out loud. A good friend can tell you, hey, well, think about the good days.
And also normally you'll be like, oh, I feel the same, and they feel the same. I'm not the only one in this situation.
Yes. And I think at the end of the day, what you will learn is that you can only control so much. The only thing you can really control is your feelings and your reaction to whatever's happening in your environment. So always try to make the best of the situation.
Exactly. And then just try to be real to yourself. So if you're just alone to yourself, just be real. What am I really feeling? Yes, I am sad, and that's okay. We'll just deal with it. If I cannot deal with it, I will deal with it with my friend together.
Exactly. I think that's also very important, because we didn't touch too much on the topic, but when it comes to working in the lab and being so busy and always running after the next experiment, the next success, you may tend to kind of forget that you have a life, and that could lead to mental illnesses, to depression. And you need to speak up. You need to speak up and say, well, I need a break, or I need to talk to someone, or I need to talk to my friends, or I don't know, I need to take the dog on a walk because I need that space.
For sure. And also that's something that I actually constantly talking to the students in our lab, or in I was co-supervisor, is that, okay, so right now it's a pandemic, and we're in this situation, and right now the most important thing is your mental health, is how you feel. If you feel in any way not comfortable or not feeling great, let us know, and we'll go through this thing together. This is the priority right now.
Yes, I think that's a great message to end on. Thank you so much, Elva, for your time, and thanks to Tillie, who graciously slept through the whole.
Accompanied me for the whole thing. But I will send you the picture of Tillie monitoring my data analysis.
Thank you so much. I cannot wait to publish this episode. I hope it'll be useful to a lot of people, a lot of trainees mainly, and I think everybody's gonna fall in love with Tillie.
Oh, thank you. You too.
Thank you so much.
All right. I'll chat to you later then.
Thank you. Bye-bye.
Bye.
Thank you for listening to this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, and Jin Chung. Don't forget to subscribe to the Dr. GPCR newsletter at drgpcr.com/newsletter. Until next time, stay safe.